Yeah, thank you for having me on such short notice, but I'm excited to share with the people here for the workshop just some of the work from my postdoc. So this talk is titled Rethinking Generative Models for Materials from Stochastic Interpolants, which is the method that we use to build one of these types of models, to sound benchmarks, which is what we sort of discovered along the way of trying to make our generative models make sense. So I guess just to tell you a little bit about me more broadly, where I kind of come to material science from is from the perspective of a computational person. And really, I would say that the unifying theme of my work, so on the left is some of my work for my PhD, but of course today I'm going to be talking to you about the generative AI stuff that I've been working on as a postdoc. But I would say the unifying theme is just this idea that we want to study materials from a data-driven paradigm. And so what that means, in my perspective, is that we want to first think about quantified representations. So how can we think about structure and order in materials through learning those representations, and then what are the statistical tools at our disposal so that we can learn more about those, about the systems we want to study, and then perhaps leverage the representations that we learn. So, so for example, your data could look like, on the left, molecular dynamics simulation, and here the coloring is telling you a little bit about what the different kinds of local bond orientational order are. And this was just using a very simple unsupervised learning method to kind of color the particles. And here you can see that just by thinking about what the data tells us, we can break the system into a very low density fluid, a high density kind of liquid, but still disordered structure. And then you can see the emergence of a crystal forming. And so that's sort of what the story is on the left side. But on the right side is a little bit different here because the data itself changes. So whereas we go from thinking about, let's say, entire simulations, we can actually just think about experimental and theoretical materials. So these are things that have either been synthesized in actual growth chambers or discovered or predicted through density functional theory calculations. And yes? Please
ask on the left, how do you get a crystal to form on an empty time scale?
You use what's a very special class of potentials, which are pair potentials, but they have multiple wells. So this, in this case, the crystal that forms is BCC. So there are probably parts of the Leonard-Jones phase space. Phase space, by phase space I mean here the phase space defined by the interaction. There's probably something you could do to a Leonard-Jones to get it to form BCC, but in this particular case, it's a pair potential that has two wells.
No real barrier, just agglomerating.
Yeah, and then in some of the potentials, yeah, I don't have any of this in this talk, but I guess I just wanted to also throw this up here so that if you are interested in molecular simulations and crystal growth, that I'm happy to talk to anybody about that afterwards. I would say certain types of these pair potentials don't even really go through a dense fluid phase. They just immediately sublimate. But anyway, yeah, it's a neat way to be able to study crystallization on sort of simulation length scales without any kind of special sampling techniques. Okay, but to sort of keep going with this story, right, so the idea here is that the data fundamentally looks different because we want to ask a different question, which is how might we design materials from the ones that we already know about? And so I want to tell you a little bit about how generative models are going to do that, but to tell you about that, I'm going to have to tell you a little bit about generative models and how they work. And I'm going to try to give you an analogy through images. So this is just a prompt I gave to DALL-E. I asked it to produce an image of UCLA, which I think is this Royce Hall? Is that what that one in the middle is called? I don't know the buildings that well. I just know Royce Hall is the famous one. But I asked it to generate an image where UCLA is on the surface of the sun, right? And it knows what the sun looks like, and it knows what UCLA looks like. It can kind of figure out something in between. I think it's more of a philosophical question and definitely something that people debate about, whether or not this is interpolation or extrapolation. Really this idea of how good are models at learning beyond what you provide them. And there's some evidence to suggest that they should be able to generalize and really kind of learn beyond what you provide. And there's some evidence that suggests that the perfectly trained models just perfectly memorize what you tell them. But I mean, so I'm going to kind of sidestep the what we actually think machine learning is able to do, and at the very least show an example that kind of should hopefully convince you that we're at least learning something that we might not have been able to know about before.
So not a serious question, but if that's on the surface of the sun, what's in the background?
That's a great, yeah, I always thought that was funny. Anytime I ask it to do this, it still puts a sun in the back. But it like still, and then it makes lava on the, so it's like somehow figuring out like hot means that the surface has to look kind of like lava, but then there's always still like weird stuff in the back. Yeah.
Does that mean it will always try to generate the material? If we look at the materials now,
Yeah.
try to think of the same thing. Does it mean that it will always generate materials that are kind of similar to what it's seen? Because it's probably seen always having sun in the background. It's never seen an image of actual sun, so it's probably going to generate again. Similar materials that will not generate what I want exactly.
I mean, this is part of the conflict, I think, of whether or not you are meaningfully learning how to generalize, or if you're just learning how to memorize, and then you just happen to be like interpolating between data that it's seen. And I can't answer that question. I think it's a sort of an open question. Something I can say about materials, we can talk more about this later, but something I can say is that, for my understanding, any new structural prototypes that are discovered through like these materials discovery methods that leverage generative models, those new structural prototypes seem to basically be known structures that exhibit some kind of basically symmetry breaking. Maybe that's just some of the models, not all, but that's sort of, I think there's a benchmark that was made by a group at Minnesota, and they're like one of the benchmarks on this page that you can look at is like how many new actual structures. And this doesn't just mean the difference between BCC and cesium chloride, which is like there's like a Wyckoff site splitting there that happens. But this actually means like a new structure, and the answer is like 0. So, but maybe that's just what chemistry dictates. Like maybe that is actually the ground truth, but maybe that might be a point for your argument that maybe you're not really learning anything new, but we think we are. So okay. So I'll just keep going so we can eventually get to the materials part. So to think about this in the context of images, what you want to do in order to make a generative model is, let's say you have a set of samples. So in this case, let's just fixate on images of dogs. This is a very like classic example. And what you're hoping is that if you give a model a bunch of images of dogs, all of those dogs are hopefully going to be a representative sample of the distribution of the images of dogs. So for example, here, there's the true distribution is like what I might call P data, and then the actual samples are like these orange dots on the distribution. And so you give it these samples, and what you're asking for is to learn more things from P data, new samples that you don't already have. The way that you do this is that you leverage a base distribution, which is easy to sample from. So often this is just some kind of Gaussian or in this case, sort of uniform noise, uniformly kind of colored noise. And so what you want to learn is the mapping from that easy to sample from distribution, the distribution that you actually want to sample from, that is non-trivial to sample from because you don't actually know what it is. And you can think of this like you are taking one distribution and you're actually learning what's called the push forward, which changes or sort of alters that distribution to match your data distribution. And this is, I think, sort of more under the purview of a technique called normalizing flows. But in our case, we kind of cheat, and instead of actually learning that mapping, we just learn the velocity field that will take you from one sample in the first distribution, the easy to sample from distribution, to the distribution of data that you care about. And so this is the idea here is that we're going to learn that velocity field. We're going to learn how to flow from one distribution to the other. And so that learned mapping is called the velocity field, and we kind of put it between time 0 and time 1. So the distribution at time 0 will be P0, the distribution at time 1 will be P1, and you could, in theory, write down a number of arbitrary different paths. So for example, you could think, so here we have these half moons that become a checkerboard, and you can see how the shape of that distribution changes with time. On the bottom, you can see something kind of similar here. It's not actually starting with a half moon, it's starting with a Gaussian, and it's going to a checkerboard. But one of these is sort of sampled using an ordinary differential equation, and the other one as a stochastic differential equation. And so you can see one of these paths or trajectories looks a lot more noisy than the other. So just a few points on this before I introduce the method that we leverage. So different paths can lead to the same final distribution from the same initial distribution. It's just the states that we traverse along these paths differ. There's no a priori best path that we kind of know before we actually apply it to the system that we're interested in. One thing that's challenging is that sort of mathematically, from a theory perspective, it should make absolutely no difference. But in reality, in the process of actually trying to learn, it does seem to make a difference what kind of flows you choose, meaning what kind of paths you choose to learn, and what kind of, let's say, base distributions you choose. So what is your easy to sample from distribution seems to make a huge impact on how well your models learn, even though in a theory sense that shouldn't be the case.
Just a question on this, because it came up in the previous thing that we didn't know the answer to it. So it's a two-part question. One is, if you have a stochastic differential equation versus an ordinary, is it the same basis of attraction? And then if you add stochasticity, you say the distribution is different. In what way?
So the base distribution wouldn't change if you add stochasticity. It would just be the path that you traverse. As for the basins that you're sampling, it's a good question. I'm not sure. I'm not sure it would change. I don't know any reason that it would, but I can't say that it would.
Because we had different views, and it seems like even the same person would give two different views at the same time. One being that stochasticity gives you more variety. The other one is that it would tend to get you more towards a global minimum, like or a bigger basin of attraction to not offend Richard. Not make it Hamiltonian, but those two seem opposite to me. As getting to a better image or a better final or a bigger. The other one is to get more variety. It wasn't clear adding the noise which one it does.
Yeah, I'm not really sure either. I think actually the paper that's actually cited here in the sort of paper that our work is based off of makes some arguments about, I think people have written or claimed that maybe adding stochasticity is helpful for generalizing. But I think that the hard thing is we don't actually know the answer. And so what we do in practice is we simply just try everything. We try a very flexible framework and we just see what performs the best. But yeah, I'm not sure. I would have to think more about that. But it's a good question. It's, yeah, I can think more about it after the talk and we can discuss. Okay, the last thing I wanted to say about this slide, and by the way, I appreciate the question and keep them, just feel free to interrupt. I'm super happy to take this slow. So the last thing I wanted to discuss is just that, so you're mapping two distributions. You have your materials data set, let's say that's the final distribution that you want to map to, right? That's where your samples are. And you're going to start from some distribution that's easy to sample. But something I think that might be worth considering is that, and this actually maybe speaks to your question, is that there is an inductive bias reflected in both the way that you map as well as the data sets that you're choosing to connect to one another. Perhaps a good example of this is thinking about machine learning or atomic potentials, which are trained using data that tends to be very near equilibrium. So just simply by doing that, it means that they show systematic softening at material that in configurations that are far from equilibrium, so they underpredict the energies of those states. And that's an interesting consequence just by the fact that the data is not fully representative. Okay. So we seek this velocity field, and there's infinitely many paths. And so the way that you might learn, actually kind of go about learning this velocity field.
Just a question on that point, because I don't get that what you said. And it seems important, is that if you fit near minima, why should it be softening as you go along?
So it's not the minima, it's when you move away from the minima.
Right, so when you move away from where you fit around
yeah.
minima, why should you know that it's systematically softens versus anything else? Like how do you know that?
I think, so this is specifically, I should have put the citation here. This is specifically referring to a paper that I will dig up for you after this that was published somewhat recently. And I think it was literally called like systematic softening in machine learned potentials. And I think what all they did is they just looked at the potential energy surfaces predicted by a machine learned potential and an DFT. And they just compared them. And that's what they saw. And they actually corrected it.
Because I would have thought as you move away, you could become stiffer or softer. Why would it favor softer? What's in?
Ah, I see what you're saying. You're saying, why is it always softer?
Yeah.
Maybe because you're always, it might just have to do with how you're interpolating between your data. Like something about the bias baked into the functions that you're using to, right? Like it's, there's some work done in thinking about, like, for example, how like a variational autoencoder might, how it bridges samples together in effect, and like actually perturbing the sort of geometry of that learned space is possible. There's a lot of, I think, I don't quite know how to prove this, but there seems to be a lot of bias in terms of how samples actually get bridged in the process of learning.
I mean,
yeah.
If you really just learn on minima, and it doesn't learn about the curvature around the minima, and it doesn't necessarily learn the energy you guys either, but then it just interpolates between them and says, these are all points, I can interpolate with them, and they all equally likely, so like the low energy, and it does never figure out that there is actually quadratic, that the minima are actually quadratic, right? Because we never gave it that information. We never gave it information, these are the real structures, these are false structures.
I mean, I get the point if you're saying you have 2 minima and you interpolate, you're going to get a lower barrier.
You essentially underestimate the barrier, and I think that's what we're talking about.
Probably what, yeah.
We also generally think that when you extrapolate, all your models just go crazy. Like,
yes.
It wasn't minus. To have them all go low would actually...
Or when we get out, yeah. But I think...
be terrible for like unsolvable.
When you look at especially the autoencoder methods, which just really interpolate, right? They make something out of it fuzzy.
Yeah, this is for MLIPs though. This is for these potentials, right? That are just trained. I mean, I think it's what the argument is that it's a reflection.
Did they know about phonons?
I don't think. I mean,
why? They don't. If you really just learn on the ground state structures.
Yeah.
And then try to figure out from there what energy forces are far away from it.
Usually you MD, right? So you have some phonons in there.
Well, I don't know what the specific paper says, right? That's a good question. If they do learn the basis of attraction, they should probably not always underestimate, right? I agree. But
we should look at this paper afterwards. I will pull it up.
A strong statement.
Yeah, it's literally the title of the paper. I think it's in like Nature Communications or something. You probably Google it right now. Okay, so just to keep going. Okay, so how are we going to actually learn this velocity field, right? So in practice, what's this going to look like? So using a technique called conditional flow matching, the idea here is that you just set a condition on the fact that always at time 0, you're going to pick a sample from your base distribution, and at time 1, you're always going to pick a sample from your data distribution. And then you're going to construct a path between those the two samples, so one from each distribution. And so here, for example, if I write down, this is a version of the path that is just a line. So, and the line meets this criteria that at time 0, I'm at my first sample, and at time 1, I'm at the second sample. And so these samples are how we're going to do learning. And what that looks like is that we basically build what's called the marginal distribution that is built on our samples. And so I think this video does a really nice job of explaining this. So basically, if we pick samples over and over and over again, what happens is effectively you do learn the distribution that maps you from P0 to P1. And the nice thing about this is that there's actually proof that if you wanted to, so if you're writing on the loss function and you're trying to have your model, which here has parameters theta, learn a velocity b. So you can train the loss function to basically minimize the error between your learned velocity and the true velocity. And the nice thing is that you can mathematically show that the gradients here are identical. So you can move from having to learn that field kind of construct this entire path, you show that you can actually do this with samples. So that's the idea from conditional flow matching, and this is a really nice guide that I pulled these videos and images from. And so this is going to basically build the language that we use in stochastic interpolants. So all that stochastic interpolants are is just a more general form of doing what I showed on the previous slides. So on the previous slide, I pointed out that the path that we chose was a line. It doesn't have to be a line. Also, you can add what's called a latent variable, so this gamma t times z, where z is sampled from a Gaussian distribution that's centered at 0. So it's a random variable, and so sort of over a lot of samples, it'll average to 0, but for each sample, it'll push you away from your path that's defined by your function I here. And so I guess I'll just jump back very quickly. So 2 slides ago here, I gave you a version of I that was just a line. So it's called a linear interpolant. Jump back ahead. But we don't have to, again, we don't have to draw a line between our samples. We can draw any path that we want. And this is the flexible framework that I was hinting at. Yeah?
If we draw a line, that would be trying to learn the optimal transport, right?
Not necessarily. I mean, that's, I guess, optimal transport in a physical space sort of sense. I mean, this is assuming that you're thinking of something very, in a very sort of, yes, I kind of agree that is the optimal transport, and it's the optimal physical path. But optimal transport people usually use to refer to like thinking about how we're going to like rearrange, let's say, which samples from the first distribution map to the samples in the final distribution. We're going to rearrange those so the paths are even shorter, right? This is not, it's not quite that, right? Here I'm not deciding, it's just how I'm going to draw the paths. It's sort of the optimal way, it's the shortest way perhaps to draw the paths. But that doesn't necessarily mean it's optimal. That makes sense? Yeah. I mean, it does, your intuition is right. The linear interpolants do work quite well. But like diffusion models, for example, don't use linear interpolants at all. They use a completely different functional form. They also don't use an ODE. They use an SDE for sampling. So they add noise. So yeah, so the cool thing about this framework is that it's so general that it encompasses 2 very popular kinds of models that we see in generative AI. One is called conditional flow matching, which is actually exactly the example I showed earlier, where we're drawing a line and we're going to do that kind of interpolation between samples. Diffusion models are sort of the other end of the spectrum. So if you used a variance exploding noise scheduler, which with what's called a score-based diffusion interpolant, and you use an SDE, you map onto a diffusion model. But those are just like 2 limiting cases. You can have anything in between as well. So that's, I guess, the nice thing afforded to us by this flexibility. Yeah, go ahead.
Why would anyone draw curved lines of a straight line?
Why would anyone draw?
Optimal. I mean, straight line seems optimal to me.
Yeah, I mean, that's like asking why would you use a stochastic, why would you sample in a sort of stochastic way? Why would you add noise as opposed to just learning?
That's also good.
I mean, it comes back to this idea that perhaps you expose your model to more. And by seeing more things that are sort of maybe further away from, yeah, it's, so I guess I'm going to give you a very specific example. So in the talk, the rest of the talk, when we get to the materials part, all of the materials work starts from a base distribution that is easy to sample from in the same way I sort of said earlier. That doesn't have to be the case. We could just be, our base distribution could just be our data distribution again. We could be bridging sample, like it's arbitrary, right? The distributions that you sample from are supposed to be entirely arbitrary. So I think this is just a way of being so general as to accommodate for the fact that, so like let's say if our initial and our final distribution were our data distribution. In that case, you might want your model to explore further from the paths that are defined between there, right? Effectively exposing it to like potentially negative samples. Does does that get any closer to potentially giving you, like, why would anyone do this? It's a good question.
For the distribution, it's more optimal if the initial distribution is somewhat closer to the final one.
It is. In practice, it is more optimal. Theoretically, it shouldn't matter, but in practice, it's often, yeah, we see that it's more optimal as well.
For the path, do you have any knowledge like that? How to design a path that's...
We don't design the path. We offer several. So actually, this is an example. In this table here, there's listed all the different sort of functional forms that we use. Yeah, I mean, the reality is that we hoped certain things would happen with certain paths, and we were sort of surprised. So for example, there's an encoder-decoder interpolant, which is actually shown on the right, the third column in the image on the left. I don't know if this has a, oh yeah, okay, you can't really see it, but this one, yeah. And so what you see is actually, it starts, so it's like we're bridging here, left side 2 Gaussians, right side 3 Gaussians, right? In doing that bridging, what this interpolant does is it takes you, squeezes you all to the center, and then it unsqueezes you back out. That's sort of the idea of this one. We thought that one would work amazing, but it actually didn't work that well. I couldn't tell you why. Yeah. I mean, the reality is we're kind of learning about how the models think, and we're trying to justify how they think kind of after the fact, because it's very hard to predict ahead of time what's going to work better, because the ways in which they work better or worse are not well understood, because again, theoretically, they should all work perfectly. So, okay. And then this actually maybe speaks to the earlier example I was just kind of giving in my answer, where you can think you don't have to pick a super arbitrary or easy to sample from base distribution. You could pick, let's say, your base distribution is your set of flowers, and the distribution you're trying to learn is a set of flowers. And here you can see this is what the different pathways might look like by just changing, let's say, the kind of interpolant that we're using, or whether or not we employ this latent variable, this gamma z. So here, for example, no latent variable. This is just conditional flow matching. Here we have this latent variable, and it means that the flower gets sort of partially noised as you go along, and then unnoised and turns into the other flower. So this is the very, very general idea here. And then the bottom is the encoder-decoder one, actually, that I was mentioning earlier. So this one goes fully to noise in the middle and goes back out. So we really thought that one would work quite well, but it didn't. We actually thought it should really kind of look like a diffusion model, but it doesn't. It just diffusion model with half the time. Was there a question in the back? Did you give a hand raise? You look like you had a hand raise, and then you put down.
Oh, sorry.
Okay. 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 So now I'm going to jump into talking about materials and how we take all of these more abstract ideas and apply them to learning about the manifold of stable materials, which is the one that we're interested in. To talk about materials, it would probably be helpful to tell you a little bit about what crystals are. So just like a little crystallography 101. So a crystal is an infinite arrangement of atoms that periodically tile space. You can kind of break down this infinite object into its small constituent, which tiles space. The field of crystallography is very good at this. So in this particular crystal, this is a clathrate structure. It has a sort of cage-like. It's very low density and low bonding around each atom. And so this is the unit cell marked in red. So if I take this little red object and I tile it around, I will reproduce my whole crystal. And so the representations we use to talk about our materials are unit cells. Here, I just have this notation, but you can kind of ignore it. I don't think it's going to be important for the purposes of the talk. But this unit cell, I think it's important to mention, it's a non-unique representation. Often, crystallographers prefer representations that contain all the symmetries that exist in the crystal. And so this is actually a bit more maximal than you need. It's a bit bigger. Yeah, so anyway, I won't talk about this too much. It'll come up later, but that's just an important thing to recognize. And crystals often their unit cell sizes differ based on how complex they are. So the bigger the more atoms you need to describe your periodically tiling unit, the more complexity there is in terms of the structure. Okay, so now I guess I should introduce the package that we worked on. And so this was done as a big team, a collaboration that Richard is in fact part of. And so this is called OMatG for Open Materials Generation, because everything that we do is open source or going to be open source. And so the idea here is just exactly as I've been saying, we start by sampling easy to sample distribution. So this would be a uniform distribution of positions. We use, for the lattices, we actually use a pretty narrow base distribution kind of taken from our data set. So we kind of make it look roughly like the lattices that show up in our data. And then the species actually don't really use our stochastic interpolants piece. These are learned through what's called discrete flow matching. But you can basically go from this. The other thing that you should notice about this, this very noisy crystal that's not particularly realistic, is that there's here we have these atomic identities on the right, and they're colored by the different atoms they are. But on the left, these particles actually have no atomic identity. So they learn to jump from their masked or no identity state into the actual chemical identity that they have. And so this is another sort of aspect of what we learn. Okay, so how do we use stochastic interpolants for this? So exactly as I said before, we're going to learn how to bridge samples. Here we're also going to actually sample time to kind of make this a little bit easier. So we're going to sample a time. We're going to sample from our final distribution, a structure, and then we're going to interpolate these two. And so we're going to regress onto the velocity using our loss functions. If we're learning an SDE, we can also learn the denoiser. And so that's, actually, sorry, this is very important to point out. The velocity here is predicted by actually like a model. So here we use, the model is called CSPNet. That probably doesn't mean anything to you. It's just a graph-based model that builds, so we're already, to talk about crystals, right? We've moved from the infinite crystal to the unit cell. So that's the step one of the representation. Here, this is step two. So we're actually going to learn the representation of the unit cell through this graph framework. What's nice about this, about these types of encoders is that they capture a lot of the symmetries that are present in crystals. So for example, our model that we use to predict these velocities is translation invariant. So if I take all my particles and I translate them, that should not change the velocity that I predict. Or it's permutation equivariant. So if I rearrange the order in which I write down my atoms, I should also rearrange the order in which I predict the velocities for each of those atoms. So that's sort of a neat detail that I think is relevant. And then finally we choose an ODE or SDE sampling. So this is really the full sort of space of what we do. And yeah, this is just a nice figure to try to show you this. And so if, let's say this gray atom is our particle from our noisy, easy to sample from crystal, and we're gonna bridge it to our purple atom, which is actually from a structure from our actual data set. This kind of shows you the design space of the actual machine learning, the actual flows that we learn. So on the left is, let's say, the choice of the interpolant. On the middle is, let's say, are you going to have an ODE or an SDE? And on the right, it's, are you going to use this latent variable, which is going to push you, with averaging to 0, push you away from your path? Okay. And again, this is only we only use this for the positions and the lattices. The species are learned in a different way. Yeah?
Just to make sure, this is not a box of atoms. Each point is a configuration in the interspace?
This is a box of atoms. We learn the velocity for each atom and each lattice vector. Yeah. And then a neat detail I'll also add is we had to think about how we could generalize this whole bridging samples and whatnot to periodic space, because for the positions, they exist in this periodic box. And so we're doing stochastic interpolants now on a torus. And we really do, I think, this is sort of the cheapest, simplest thing we could have done, which is that we We figure out what let's say the geodesic is this blue line, but we want to, let's say, use a different interpolant, so not the linear one. It's actually going to be this path right here, but because this goes out of the box, we're going to like take it and then we're going to bridge it back in. So this is just kind of trying to show you how we handle the periodic boundary conditions. And you can see, let's say this is done. This is using a latent variable. You can see actually the average here still collapses onto this. In this case, it's the linear path. But we started here and we ended here, and the geodesic has to leave the box and come back in. All right. So now, and then I also want to introduce to you the two kinds of models we build. So we have all that flexibility to design models. So what we can do is both what's called crystal structure prediction, which is that we actually don't do any learning on the species in the flows. We do expect our model, our encoder, to learn about chemistry. But what we do is we just we have structures that already have the chemical identity specified, and these we learn how to flow these to their final configurations. The other kind of model we learn is actually going to learn the species as well. So that's the example that I showed in the image earlier, where you can start in these random mass states and then jump into your chemical identity. So these are the two kinds of models. They're useful for kind of different things, right? The first one is useful for figuring out what a valid crystal structure might be for a specific chemistry. And the second one is useful for predicting new kinds of materials and potentially also new chemistries. So the first one can't do that. We have some relevant data sets in the field. The first one is called carbon-24. This is 10,000 carbon structures that are relaxed with DFT. I guess this is basically a toy data set in a way. And the reason it's a toy data set is because you can kind of bypass having to learn about chemistry because everything is carbon. So this is really kind of testing how much you can learn about structure. The second data set here is what's called Para-5. It is all like perovskite structures, and they are in these kind of cube-shaped unit cells, but they're orthorhombic. An interesting, I think, feature of this data set is you can really kind of bypass learning about unit cell equivariance. And the reason I say that is because all the unit cells here, the shapes look the same. And so you make it very easy for your model to figure out what kind of lattice it should learn, because it's basically pre-prescribed from the data set. And then the third data set that's relevant is probably the most general. This is from the materials project. It's 45,000 structures. They have a cap of 20 atoms in the unit cell, but they're very much realistic structures. A good amount of them, I think, are also from the ICSD, so they're experimental structures that have been relaxed with DFT. Yeah?
What do you choose to train on, like max 20 atoms? Why not?
It's a good question. Yeah, you can have more. It gets worse as you have more, like the quality of the model. It's harder. I will show you a little bit of stuff near the end of the talk that might speak to that. But this is also just a very standard data set in the field. Yeah. I've been seeing some like MP60 around. There's another one not mentioned here. It's called MPTS 52. It has 52 atoms as the maximum in the unit cell. But something kind of interesting about it is that the TS means time split. So it's actually put in order of like when the structures were discovered. And I actually think that makes it a lot harder as well. If you don't time split the data set, it works a lot better. But it's like, by definition, it is split in time. Yeah. So I don't know if you're familiar at all with like the standard data sets in the field, but that's just some insight that I came to recently. Okay. So for crystal structure prediction, right, the way that we benchmark it, yeah, go ahead.
Sorry.
No, it's fine.
You said time, that was interesting. Do you mean like if we use only like before a certain time, like if we use all the materials that were invented before a certain time,
Yeah.
it's hard to predict the future?
Yeah,
like
I
stock
think
market?
that's why they did it like that. I think yeah. Maybe it speaks to the ability to test like how we generalize, how models I don't really, yeah, it is strange though. It's like you're hiding information from the model that could be really useful for it to learn stuff. But it's still, yeah, so the numbers for that data set are a lot worse in terms of benchmarks and performance. And for a long time, I thought it was because of the 52. And now I think it's because of the time split. So that's sort of a newer, it is just something I learned very recently, yeah, by playing around. Okay, all right. Okay, so crystal structure prediction. I just want to say a little note about benchmarks, just because I'm going to talk a little bit more about them. But the way that we might benchmark these models is relevant in how we train them, right? As I mentioned, we have this very flexible design space of interpolants, and how we're going to train our models or figure out which ones are the best are through benchmarks. And so the way that we would benchmark crystal structure prediction is we take a test set structure. In this case, it's gallium telluride. And then we start it with a random initial structure, and then we're going to evolve it forward with our model. Right? We've trained the model already. Now we're going to do inference. And then we're going to see, did this first structure, right, this thing that we predicted, is it going to match the one from the data set? And we're going to use this thing called structure matcher, and we're going to say, okay, yeah, this was a match, and then we're going to assess the quality of that match, which is the root mean squared error between the sites. It's this number. I can just tell you kind of qualitatively that's a good value. Anything in the tens digit is bad. In the hundreds digit, it's good. Okay, so that's how you would benchmark crystal structure prediction, and we basically, computer scientists love tables, and so we make these tables to show how we do on benchmarks. I don't want to talk about this whole table. It's really quite a lot of numbers, but I think what's just cool to point out, and I already got asked about why might you want different interpolants. I mean, some people think like diffusion models are the best, and some people think conditional flow matching is the best. We just didn't assume any one way or the other, and we kind of put this to the test. And some of our best performing models were with the linear interpolant, but also with SDs. We also use this trigonometric interpolant, which actually takes you a quite weird path, I would say. But yeah, so what I really this is too many numbers, but really I just want to point out this is perhaps the best performing crystal structure prediction model we had. Still like roughly 30% unaccounted for. So we're hitting around 70 in terms of how much of our test set can we reliably repredict. That's kind of the question that's being asked. Okay. And then with de novo generation, the primary benchmark that we think about is stability, uniqueness, and novelty. So it's called a Sun score. And it just differs, again, in that we're learning about the species. So we need to evolve the species during the flow as well. And here I just have an image of how we, let's say, construct the convex hull. So when we assess stability, we're thinking about being within 0.1 EV per atom of the convex hull. And this is important because if we're pretty far from the convex hull, what's probably going to happen is that you're going to decompose into effectively phase separate into different compositions that are lower than you on the convex hull. And so that's why being close to the hull is very important. And actually that's why it's convex. It's just that if you were above, you'll just end up phase separating. Uniqueness just has to do with when we go to generate structures over and over and over again, how many of them are just the same thing. So we would like to maximize uniqueness. We would like to have as many different answers as possible. And then novelty is just, are you regenerating structures you already knew about, or are you discovering new ones? So the rate of, let's say out of 10,000 structures, your Sun rate is going to tell you just how useful your model is for predicting new kinds of materials. And just to kind of illustrate, again, sort of the process through which this is done, let's say you have a test set structure. Again, we're taking this gallium telluride, and we randomize it. So we randomize the positions and the cell, and then we already have a trained model, right? This is, again, just the inference part, and we do what's called de novo generation. And then we actually add an additional step here, which is relaxation. We either use DFT or a machine learned potential for this. And you'll see here, of course, we're a very different chemistry than what we started with in our test set, but that's fine. We're not evaluating whether or not we predict, we don't care if we make the same chemistry. We just want to see if it's stable, unique, and novel. It's actually not stable. So this particular structure would not sort of pass our sun score test. We also, so in training, I guess a sort of subtle point here is we can't really train on sun scores because they have, as like a benchmark of interest, because it's very expensive to do DFT. So we actually, the way that we assess the quality of our models before we go to predict the sun scores is by looking at a number of benchmarks having to do with the distribution of things that we generate. So for example, one thing you might care about in terms of asking, do the materials I've generated look realistic, is that you might think, let's say, how many unique elements are there? And so we know, let's say for existing materials, roughly what that distribution looks like. So we would actually like to get very close to what the existing distribution is like. Of course, if it's too close, if it's too perfect, you might not generate any new materials. So it's an interesting trade-off between wanting to get very close to your data distribution in terms of what you're generating, as well as being able to extrapolate and learn about new materials. So here, just on the left side, is a different kind of competing model. It's a diffusion model for crystal structures. And then this is our model. And so here, you would see between the gray and the purple, a better match, for example, in the unique element count. The bottom here is actually a benchmark that we contributed. This is the average coordination number, where coordination number is counted as the number of bonds for each atom in your crystal, and you average over this number of bonds. So the average coordination number just gives you some kind of fingerprint of a structure, and you can actually plot this distribution out, and you can see that where, for example, where diffCSP might suffer a little bit with predicting these, like, let's say, high coordinated structures, our model does a little bit better there. So this is just another I think we were excited about contributing sort of additional materials-informed benchmarks for this. And then finally, to show you our results for the de novo generation, we achieve, at least at the time, state-of-the-art performance with our model, and I think this is partially owed to the flexibility of it. And just to quickly kind of touch on sun scores, I mentioned it's basically like the intersection of all the stable structures, the novel structures, and the unique structures. But what you might notice, so here we actually have plotted the novelty rate in one column. So if you look at these like kind of right three columns, we have a novelty rate and a stability rate. And there's definitely a trade-off. So things that tend to be very novel are less stable, and vice versa. Things that tend to be very novel tend to be less stable. Of course, this is kind of the natural kind of competing forces that are happening when you're trying to discover new materials. But we show that our for the naysayers in the back, the linear model with the stochastic differential equation and a latent variable was our best performing sun rate model. Yeah.
You don't have linear OD here, and the last one you did.
That was for, yeah. So that was crystal structure prediction.
And there it was winning, right? Linear OD was the best one.
Was it? It might have been, yeah.
Yeah. So.
Yeah. But this one, yeah. All right.
I was wondering if you could just, summarize or give any sort of intuition for what knobs you have to go between, say, stable and novel or unique. I mean, apart from just trying things or sampling hyperparameters or whatever, like, if you add stochastic terms, how does it change it in a systematic way? Like, what knobs do you have to know, if any?
That's a very hard question. I mean, I would like to be able to answer that question. There are a lot of hyperparameters. A lot, and
some insight.
So I will say it's been a little bit of time since I thought about this, but I was hunting for this. I was looking. So we actually publish all of our hyperparameters for all of our best models, and there are trends in there. Like something I remember. Oh my gosh, it's gonna be hard to remember off the top of my head just because there's so many models, and it's like kind of juggling many pieces, but. for example, some of the, it's like there's a balance between how much of the loss function. So the loss function is actually like kind of summed over the different pieces. So you have the positions, you have the cell, you have the species. But so let's say between the positions and the cell, actually kind of interesting is that balance. Yeah, just so for in some cases, the models would put all of their effort into, so this depended on the interpolants, right? So sort of given a set of interpolants for, let's say, the positions, I can go look at what were the hyperparameters that were chosen. And so there were some trends. I can try, I think we actually did summarize them in the appendix of our paper. It's interesting to me as well, because I would also like to understand how the choices we're making affecting the ability for the model to learn.
Is there any takeaway that you remember, like tune something in some way, get more unique?
Yeah, I think, unfortunately, a lot of the takeaways that that I remember, like one that I fixated on quite a bit was unit cell equivariance. But what we realized, sort of in hindsight, was actually the data set was the problem, not the model. So I was trying to fixate on why, for example, one might want to use an interpolant that takes you very far, a path that traverses a long way as opposed to a short way. Yeah, but the one that really jumps to mind, the one that I still remember that I think is true, is relating to the weighting between the learning of the positions versus the learning of the cell. And so based on how much, if you do I think it's like if you do an SDE, or if you have a very nonlinear path, or use a latent variable, I think it tended to weight it would often like throw all of its weight behind positions. But then in these, somehow for these very specific cases, it would do the total opposite. It would throw all the weight behind the cell. But I would have to like look at those tables again to answer your question. But I think we did actually try to write about this in our appendix. Yeah.
I have a simpler question. I would like to go to the point before that about the average coordination number.
Yeah.
Is this a unique qualifier? I mean, of course, when you have
no,
you need a distance criterion, some so to say. So I wonder whether it's then useful if it's not really unique.
So it's, I think so this is just the distribution I'm showing. What we actually do is that we take the Wasserstein distance of this, so we basically are trying to map, so we have a purple, it's kind of hard to see, but there's a purple curve here and a gray curve. And we take the Wasserstein distance, which is the Earth mover's distance between these distributions, and the closer they are, the better, we say the better our model does at recreating the data set. Yeah, or the patterns in the data set. Yeah, and as far as I know, we are actually still the best model for that, even compared to some symmetry constrained models, which you would think would do better. But I think I just saw a paper today that I think we're still sort of, for whatever reason, our models are still better. So, but yeah, the main thing I noticed, really, the reason I thought this was relevant was actually for these high coordinated structures that live out here. The fact that, for example, one might be bad at predicting like an average of like, I guess in my mind, I mean, things seem a lot more prototypical to experiments. Maybe like if you do experiments, certain kinds of structures might seem more prototypical to you, right? So I looked at this, I was even just shocked the fact that it wasn't like dominated by high coordinated things, but it's not, just because the data set is diverse by design. But I was figuring, like, if you can't produce high coordinated structures, I'm not sure how good your model, like if your model's worth anything. Actually though, if I can see it on the screen, it may be a bit hard here, but this model seems to do, the one on the left does better for low coordinated things, but we do worse for whatever reason, but yeah. Okay, how much time do I have left?
Technically 3, 4 minutes, 10 minutes or so, but
10 minutes, okay.
Yeah.
Well, there's like basically a whole second half of the talk that has to do with benchmarks, just enormous, what I would call like carelessness in data set creation and benchmarking.
I would love to hear about that, by the way.
Okay. Well, I'm happy to keep talking. I'm also happy to pause and just talk to people about that privately. I'm also happy to take questions.
You okay with talking a little longer?
I have to leave early. So.
Okay. Most people are okay.
All right.
Axel will have to. I think
I
it's
have to
recorded.
I have to pick
you can
up
watch
class.
it. Oh, you can leave. Wait. Thanks for doing that. Yes, so keep on going.
All right, I will keep going, but
Please speed up a little bit.
I will try to
the highlights.
Yes, I will try to give the highlight reel. And if you need to go, please go. Yeah. All right. I will. Yeah. And I guess just save your questions, and I'll try to go fast. Okay. So So this was the work. It was exciting. We were trying to understand why interpolants work better than others, but then we ended up getting distracted with thinking about data and benchmarks. And so, okay, I'm just going to skip this, but the point of this slide is to show you that metrics matter, because as I cited before, we choose what our best models are. We push our hyperparameters to directions that meet our benchmarks. So if our benchmarks are bad, we're going to learn bad models by bad hyperparameters. So I mentioned before, so the rest of this talk is just going to focus actually on crystal structure prediction, which was that first type of model I mentioned. I mentioned that what we do is that we take a test set structure, we randomize the positions in the cell, and then we flow it forward and we predict, let's say we predict the correct structure, it matches, yay. It turns out that you can also, let's say, predict a polymorph of that structure. And if it just happens to not match the particular test set structure that you've chosen, you penalize your model by saying, well, you didn't match. So this is quite bad, right? This seems like a very enormous oversight. And we kind of realized just how enormous it was. And it really, this is data set dependent, but I will show you that now. So I guess just a few words on kind of what helped us go in this direction. I mentioned this Perovskite 5 data set, this one that had all cubic lattices, and they would have 5 atoms in the unit cell, hence the 5, or sorry, not 5, I guess 5 atoms, yeah, 5 atoms. And what we notice is this really, really big trade-off between our quantity of matching structures and the quality of them. So if you look, let's say on these numbers here, we're in like this 74, 73, these are all the non-linear ODE models. And the linear ODE model, it would not push further. It was like pretty much stuck around 50%, but it had a very good quality of matches. So you can see like this is not precisely an order of magnitude, but roughly an order of magnitude worse. And we would go look at what we've generated, for example, here on the right, compare it to this test structure, and I did my best to kind of try to align these crystals for you so you could see where green goes with green, brown goes with brown, blue is totally off. These are a crystallographer would not call these the same structure, right? But this does. Okay. So this is kind of what took us in hunting for what was happening. This should be an easier data set to learn, by the way, than the big materials project data set. And so what we realized is this data set is just full of polymorphs that for some reason, I guess no one had noticed or thought about. And so here is a bar representing all of the structures in the data set. There's about 18,000. So in this little blue sliver are the compounds that occur one time. And then by what you see here with this teal bar, which is most of what is colored in right now, you can, these together, this teal bar and this little blue sliver, tell you all the unique compositions. And so if you're following what I'm saying, you'll naturally glean that what's here is a bunch of non-unique stuff, which is the same composition as what's here in the teal. Meaning roughly half the data set is structures that have the same composition as another structure, but of a different arrangement. These arrangements come in a few different flavors. So you have polymorphs that have distinct structural prototypes. Really what this is it looks like distortions. So for example, this one on the left and the one on the right are both in the data set. But here you can see that I think these red atoms are probably oxygens. They're quite kind of pushed to the side. They're not really symmetric about the central atom in this unit cell. You also have polymorphs that have the same structural prototypes, but you have this site swapping between atoms. So this is a huge problem, right? No one, we're basically feeding the model. Oh yeah, and okay, let me say this first. So the data set is 18,000 structures. It's typically, so for training what we do is very standard. You will split your data set into training, validation, and testing. The validation set is like used in the process of training, but is not, but is only used to validate how you're doing. The test set is how you actually go to benchmark what your model has done. It's filled with structures that you've never seen before. And so what's happening is if you take all this data and you split it randomly, is what it means is you're exposing your model to a bunch of polymorphs of, a bunch of compositions with a specific accompanying structure. And then at test time, it's going to see those same compositions, but different structures. And it's going to be penalized if it cannot predict this different structure. Okay, so we thought this was very strange. It was a very strange way to do crystal structure prediction. So we need new benchmarks and new data set splits. Right, so how do we adapt the benchmarks, right? Basically, I'm not gonna talk through this whole slide because it'll take a while, but to summarize, instead of saying I'm gonna take a test set structure, I'm gonna do inference with my model, and I'm gonna see if the structures match. Instead, what you do is you're gonna do that a bunch of times, but you're only going to be concerned with if your reference structures find, basically you're going to look for matches with respect to your reference set instead of your generated set. In effect, the idea here is to make sure that, let's say I have two polymorphs in my test set. I just want to make sure I generate both of them, but they don't have to be exactly one-to-one. I can generate them in the wrong order, and that's fine too. Okay, so that's the sort of main takeaway here. And then we also wanted to adapt our metrics to focus both on quality and quantity instead of having separate ones, right? This like match rate and the quality of the match with the root mean squared error should really just be one metric. And so we do this by just replacing, anytime you fail a match, you kind of get like a none entry, you get a none response, like a null response. And so what we do is we just replace this null response with the maximum tolerance used by our structure matcher. So it's a way of kind of trying to bake in a little bit more quantitative information, and then you can kind of combine the metrics. So this is what we call this is corrected RMSE, because what you're basically doing is you're measuring the RMSE between your generated structure and your test structure. And instead of reporting a null result when you don't get a match, you just report a number that is a tolerance that's so high that it would report a non-match. Okay. Skip ahead. I do want to show you what happens. Oh yeah, so and then we resplit the data sets. This is an important point as well. So okay, we have like the same compositions are showing up in different pieces of our data set with different structures. And so what we do to alleviate that is we force the polymorphs to be together in whatever part of the split that they need to be. And this means that when at test time you go to see, can I predict the right structure, you're seeing compositions that you've never seen before. And you have to predict both of the polymorphs. So what we see here is so this is our old results, and these are the new results. So I'm going to put stars here on these two models because we did not change any hyperparameters. So this is like, if you want the most apples to apples comparisons, you would look at these top two rows. So this is our adjusted match rate, and this is our corrected RMSE that I very quickly mentioned. You can see this match rate jumps up by about 20% from these models to these models. Again, same model, just trained on the same data, split differently. And this corrected RMSE also improves from this one to this one. And then here for our models, we went a step further. We actually optimized our hyperparameters differently. Instead of optimizing for match rate, we optimized for our preferred metric, which is this quality plus quantity metric, which we called corrected RMSE. And so this also led to pretty significant model improvement. In fact, the best results for corrected RMSE were predicted by our models, which is not surprising because we tuned them for that. Okay, very quickly, the last thing I'll note is that another oversight we noticed is that we have crystal structure. People have been doing this for a while, like before us, making crystal structure prediction models for, let's say, the carbon data set, which has all carbon structures. And that's really also was sort of alarming. We were wondering why they were why you would expect to do a one-to-one mapping if you're going to just generate with the same chemistry constantly. So that was weird. So that was one. We also kind of started digging in that direction. We also, though, noticed that there's a lot of replica structures in this data set. So basically, I mentioned at the beginning, or near the beginning of this talk, that you can have the same crystal but different unit cells. That just happens quite a bit in this data set. And I think that's because after these data sets were probably reported with like DFT level accuracy, which is far further than what we need and what I think a crystallographer would reasonably call the same structure or not. I mean, if I can put these on top of each other and they look the same, to me they're the same. And what we noticed is this even happens for different numbers of atoms. So you can, let's say, plot the radial distribution functions. For these three crystals, they're all the same. So what we noticed is that this data set is like maximum 40% unique. So we pruned it of that. Yeah.
That's crazy, right? That's just an error. It's not due to DFT or anything.
Well, yeah. This data set is from Chris Picard's group, I think. It's actually much larger. So the original is like 100,000 structures. Then the 10,000 that it was originally made with are like, I think it's like just everything below a certain energy cutoff. And then no one looked further. That's why I'm also saying it's maybe a DFT level accuracy thing.
Oh, why? It's matching different structures that are identical with periodic boundary conditions.
Yes.
You have to have that.
I would agree. I mean, if you're matching them with a tolerance that is extremely low, you won't match them. But that tolerance is unreasonable for what we need. I mean, I'm with you. I think it's ridiculous as well. Yeah. So the paper. I don't have the figures here, but the paper actually shows how we use structure matcher to do this. We basically took our data set and we just did structure matcher between every pair of structures, and then we found, we basically plotted the boundary between when you go from matching to not matching. And the boundary, there were huge peaks at extremely low tolerance. And that's how we figured this out. So it was pretty shocking. But cool fact is that you can now leverage all this duplication you can leverage. So we made what are called overfitting. We not only pruned that data set and retrained models and benchmarked them, we also used these replicated structures to make new overfitting data sets. So this one I called carbon X because X here, we typically use X to talk about fractional coordinates. So here, this is a data set with 480 identical structures, and all the atoms are just translated.
They're all from the same data set.
They're all from the same data set. And the structures are
inflation, right?
Yeah. So the cell shape is the same. There are rotations of the cell, but the cell shape is the same too. So that's why it's just X. We made another one that's called carbon NXL. So here we actually see the number of atoms in the structure change. So of course, sort of by definition, then the size of the cell would also have to change. So that's why it's NXL. The unit cell sizes go from 6 to 16. And you can see, I think this is sort of remarkable. It's all the same crystal. Let's not forget that, right? But here we do quite well. So this is carbon X. We do 100% matching. We have a shockingly low root mean squared error, which is not surprising. Once you move to this more, we have different sizes of unit cells and different shapes of unit cells. Now what happens is we start doing a lot worse. We get like a 60% match here for just the n equals 6 atoms. We get a 39% match for n equals 8. And also between n equals 6 and n equals 8, we go down an order of magnitude in the root mean squared error.
Can you remind me in the process here, so for each sample in your data set, you scramble the atoms and then re-relax it, like the crystal structure?
That's right.
Okay.
Yeah.
I wonder whether this is a chemical reason, because with carbon you have sp, sp2, sp3 hybridization, so you have a huge variety of possibilities. And we once tried to cooperate with somebody getting a machine learning potential, and it utterly failed because it can't really reproduce this chemical variety of this. And I wonder whether this might be a reason here. I have no idea.
It might be. I mean, the model is learning nothing about so it's the chemistry is sort of like forgotten here,
Sure,
right?
yeah,
because
sure.
it's all carbon, but
it's a structural complexity, or
yeah.
I mean, and they are close in energies and so on. So maybe this makes it.
That is definitely. I mean, that's definitely true, given just how many. I mean, just how diverse. I think like. Just how many like well-known allotropes of carbon there are, and then also on top of that, just how much diversity like you still see in the data set, even after pruning away the 60% of structures that are just replicas. I will say I don't, off the top of my head, actually know what these structures are. Like I should like plot them, but I don't actually remember what they look like. I will say that just perusing through the data set, a lot of the structures, and I will say that these are, to me, still meaningfully different. They do just look like distortions of the other ones, right? Like, say you have like some motif and it's just like squished, and yeah. So that's there's a lot of that. But those we count those as distinct, right? That's when I say we're pruning away replicas or duplicates. It's those still count as the same, yeah. Yes, and okay, and I mentioned that so we do prune of duplicates, and then we also, for fun, we make this increasing or decreasing by unit cell size data set split. And so this won't surprise you, as if you train on the big unit cells and then you ask it to predict for the small unit cells, you do much better. But the reverse is much harder. So if you train on these small unit cells and you ask it to predict a big unit cell, it does poorly. So, I mean, really the takeaway that we have from all of these different fun overfitting data sets, is that this dimensionality set by your number of atoms is sort of a significant barrier. And we've, at least in these two cases, we've sort of taken away the complexity of the structure, even because you just have to learn one structure. Yeah. Versus here, you're still learning a diversity of structures. Okay, yeah, I guess this is like my final slides, final takeaways. I mentioned that we looked at polymorphs in this perovskite data set, and just how that basically half the data set was polymorphs, or had a polymorph. The same is true in this better-known MP20 set, as well as a much bigger version of it called Alex MP20. It's not quite as bad of a problem. So here, instead of having like, let's say, 50% unique compositions, it's more like 80%, 82, 80. And so we have actually gone forth and resplit these data sets, grouping the polymorphs together. And the results aren't here. They're in the paper that we have online on the archive. But you can see that it does, just by doing that, you can improve your models by just a few %, at least for MP20. Sorry, at least for this one, the one on the left. But you can see the problem is much worse on the right, actually. So that one and those results kind of yet to come. So yeah, I guess my takeaway is the data set, how you split your data, how you handle polymorphs, and how you incorporate the existence of polymorphs into your benchmarks seems like a really important thing for materials, and we should be doing this for any model, not just crystal structure prediction, right? You're going to take those data sets for your de novo generation models as well, and the same splits, and yeah, the benchmarks will change, but the data sets won't. So that's very important, and we should not be ignoring that. Okay, finally, I just want to give a little bit of an outlook slide. What can our generative models not do particularly well yet? Basically, like, where do I think we should be going? I would say large unit cells are quite interesting to me. Complex structures, things that have hierarchy in their assembly, things that really like have much simpler building blocks that then together arrange to make much, much more complicated crystals. I think that should be in the purview of generative models. We should be thinking about structural disorder, so symmetry breaking, but as well as chemical disorder, so alloys. I mean, people have started to think about this, but these are very hard problems to incorporate into the framework of generative models. And then to make the problem a lot worse, you could try to think about things that are aperiodic, or let's say you can, these twisted bilayer superconductors are periodic in each layer, but if you twist them at exactly the right angle, you can no longer describe their tiling periodically. So all of these, I think, should be under the purview of thinking about generative models. So with that, I will conclude. This is a picture of my group. So thanks to the people I've worked on this with. Richard, who's obviously here. There's a slew of other PIs, so it's quite a big collaboration. My advisor, Stefano, right there. Philip and Tom were co-authors on both of the papers I discussed today, as well as Eric, Zarin, Amit, and Pawan were co-authors on the generative model work, the actual building of the model. And then if you want to look at our code, this GitHub link is here. And so with that, I'll say thank you for listening. You've been asking questions, but I'm happy to take more questions now. Thanks.